Hi, and welcome to episode 51 of the Breaking Bio podcast. I'm Morgan Jackson, a PhD student at the University of Guelph in Ontario, Canada. As always, we've got a special guest with us, but first we'll go with our regular co-hosts.
Hi, I'm Heidi Smith. I'm a postdoc at the University of Texas at Austin.
Hi, I'm Stephen Hamlin. I'm a postdoc at the University of New South Wales in Sydney, Australia.
And as I mentioned at the top of the podcast, we've got a guest with us this week. Today we're joined by Dr. Karen James, who's a researcher at the Mount Desert Island Biological Laboratory. Karen, you want to say hi and tell us a little bit about what you do?
Sure. As you said, I'm at the MDI Biolab, which is the short version that's not such a mouthful, which is located up near Acadia National Park in the state of Maine. Some of you may know where that is. I am a first-time NSF principal investigator at the lab. I just started a year ago next month, and I work on a combination of DNA barcoding, or DNA-based species identification, and citizen science, so the public participation in scientific research, to try and engage more people more successfully in big biodiversity monitoring projects. I am also a director and co-founder of the HMS Beagle Project, which is a UK-based charity that is aiming to raise money and rebuild HMS Beagle and sail it around the world with scientists and teachers and students and all kinds of science enthusiasts aboard, and to create more science enthusiasts. And then finally, I also used to be a postdoc at the Natural History Museum in London, where I did a lot of Charles Darwin related projects, and we might get to some of those. Oh, and last but not least, I applied to be an astronaut, and I think we might be talking about that too.
Okay, look, before Heidi jumps in, I just gotta say, I feel completely inadequate now. What? God damn.
Yeah, it's true. Someone asked me the other day, like, what my hobbies and side projects were, and all I could think were, like, the side projects that I have, like, in lab, and I thought, I have nothing else. And you have, like, so many high-quality facets of you and cool things, and it's not like...
Keep it coming.
Yeah,
well,
it's
listen,
amazing.
when I had just started as a postdoc, I didn't have any of these side projects, so...
Yeah, so can you tell us a little bit about your path? Because you were like a hardcore geneticist before. This is how I know you.
And I'm not anymore, is that what you're
Sisters,
saying?
no, but like sisters of genetics forever, you know?
So I did my undergraduate work at Colorado State University, where I started working on fruit flies in a developmental genetics lab there. And then I was very interested in genetics from that, and so I went on to do a PhD in genetics at the University of Washington in Seattle, which is now, the department is now called Genome Sciences, not genetics anymore. And some of us in the old guard like to say that we got our PhDs in genetics, not genome sciences. And when I was there, I was working on Drosophila, or fruit fly, developmental genetics and cancer genetics and such topics, but I also got interested in evolutionary developmental biology, or evo-devo, which is how development has been tweaked through evolution to create all of the diverse forms that we see around us today. And because of that interest, I applied for a postdoc at the Natural History Museum in London. And actually, I switched from fruit flies to plants at that time. I moved over to the botany department and became a botany researcher at the museum. And I worked on a whole host of systematics and taxonomy and evolution projects. It was kind of a grab bag. I was the genetics person, and I was coming into a place where not too many people did genetics. I mean, quite a lot, but still I had a certain level of training that might be applicable to interesting projects. So that's what I did. And then while I was there, two interesting things happened. First is the whole DNA barcoding movement started. And that started in about 2003. And the Natural History Museum was sort of an epicenter of that. And so I got interested in using DNA to identify species, and that seemed to fit well with my genomics background and my new interest in evolution and systematics. So I started working on that more. And then the other thing that happened was that Charles Darwin turned 200 years old while I was there. And so I did a secondment from my postdoc, and I was the coordinator for the science program for the Natural History Museum's Darwin 200 campaign, which was a huge national and international celebration of Darwin's birthday. And I coordinated a whole bunch of projects, and I got to do some exciting things like go to the Galapagos and extract DNA from some of Darwin's specimens. And then
Wow.
I know, I hope we get to talk about that some more because it was really wow. And then and I also got involved in the Beagle Project during that time, which now maybe that makes a lot of sense. I was interested in Darwin and stuff, and so the Beagle Project makes a lot of sense. And then I got this opportunity back in Maine to become a staff scientist at this small research institution, the MDI Biolab, where which is just right on the doorstep of a national park. And I'm a big outdoors person, so this was like Mecca for me, and also for my partner, Bill, who is a mouse geneticist. The Jackson Laboratory, which is one of the world's preeminent mouse genetics research institutions, is also located on this tiny rural island with a national park on it. So this was a perfect place for us, and we feel really fortunate to be here. So that's my story.
For those of the people who don't follow Karen on Twitter just yet, you should, just for the pictures that she posts from hiking.
Oh, thanks.
Yeah, it's amazing. I'm like, at least I don't live in New York City anymore, so it's a little bit better. I'm like, I see beautiful stuff now. It's fine. Because it seems like the Darwin stuff sort of brought you into this more public outreach and interacting with the thing. Would you say that's fair?
Yeah, that's fair. It started with blogging in association with the project back in 2006, 2007. And that got me interested in writing for generalist audiences, and that got me interested in outreach and engagement, but then also the actual Darwin campaign, and just working at a natural history museum, public outreach and engagement is kind of a big deal. So those things combined sort of propelled me into my interest in reaching out to non-scientists.
Now, because you're so pro-Darwin, do you feel like when Alfred Russel Wallace gets so much attention, or you're like, no, that guy's done nothing, or do you not have a strong feeling?
Yeah, I totally think that Wallace is a nobody. No, of course not. Yeah, I mean, I'm a Wallace fan. In fact, I was part of a small team at the museum that developed a special Wallace exhibition during the 2008 celebration of the 150th anniversary of the Darwin and Wallace papers being read at the Linnean Society in London. I'm a big Wallace fan. Just because I celebrated Darwin doesn't mean that I can't also celebrate Wallace.
Okay, just
sorry, you got to pick one around here.
This is me rolling my eyes.
Or better yet, if you rebuild the Beagle, have them fight on the deck,
Oh,
yeah, well, I actually wrote one of my very first blog posts way back in, I think, 2007 was The Ship's on Fire, or Why This Isn't the Helen Project. Because Wallace's ship was called the Helen, and HMS Helen, and she burst into flames almost back to England after he did a bunch of collecting in South America, and he lost all of his specimens. And so then he went back to the Malay archipelago and started collecting again. So one could suggest that if that hadn't happened, that he might have had evidence for natural selection earlier than he actually did.
Yeah, that's great with the outreach. So with your outreach endeavors later on in your career, did this kind of lead you to wanting to do some of the citizen science stuff?
Yeah, sort of. I mean, I would really like to be able to say that my interest in improving the world and appreciation for science is what drove me to do citizen science. But the truth is, the truth is that, so in 2009, I was involved in a paper that was published in PNAS, the Proceedings of the National Academy of Sciences, for people who aren't sciencey. And that paper was about announcing a pair of gene regions that could be used for identifying plant species by DNA. And after that happened, we were all really interested in stopping the process of picking and choosing the method that we were going to use and getting started on projects where we were using DNA to identify species for interesting, purposes. And so we had this big idea that we were going to make a database of DNA sequences from all British plants, and that we were going to use this for all kinds of interesting research and education and outreach applications and forensics and everything else. And no one could get funding. It was dismal. I mean, we had the Natural History Museum, the Royal Botanic Gardens Kew, and the Royal Botanic Garden in Edinburgh as a consortium, and we could not get a grant funded. So if those three entities in the UK cannot get a grant, you're just not going to get a grant. So
that's incredible.
Yeah, it was tough. And I was coordinating the proposal effort, and it was just, when we didn't get it, it was just, sort of 2 years down the drain. Anyways, so I still wanted to do this project, and I happened to know through the Beagle Project someone who was who headed up a charity that ran 7 schools in the UK and 1 in France. And he found out about my interest in DNA barcoding, and he said, hey, if you will share your research with my students, I will be interested in funding your project. So I partnered with this educational charity, and we started this program working with 11 to 14-year-olds in Dorset, which is down near Lyme Regis, where Mary Anning collected all of her very cool fossils. And we worked with these kids to do natural history, collecting tree specimens, extracting DNA, making proper herbarium specimens and sheets, and doing the whole DNA barcoding, the whole bit. And this worked really well. I started realizing that a partnership with education and involving people in science could be beneficial to both outreach and to your scientific aims. And so when I came back here to the US and started working at the MDI Biolab, I was really interested in pursuing that more. And so my proposal to the National Science Foundation a year and a half ago, which was funded, was to combine DNA barcoding and citizen science, but this time working with adult volunteers instead of children.
Cool. So what's this new NSF project going to be aiming to do?
We are, you'd think I would have this on the tip of my tongue by now. So what we're doing is we're doing a pilot project to explore this idea I was just saying, which is involving citizen scientists in answering big biodiversity and ecology questions with the help of DNA barcoding. So by that I mean, when you go out to answer a big question in ecology, for example, how is climate change affecting the biodiversity of Acadia National Park? There aren't nearly enough scientists to do all of the field work needed to answer that question, but citizen scientists don't have the training in specimen identification to be able to answer that question. So you're sort of at this impasse. And the idea is that what if you could use DNA barcoding to validate the identifications that are made by citizen scientists so that they would know that the work that they're doing isn't going to waste. And the scientists who are interested in the data that they produce would know that the data are valuable. So this new idea or concept. We are piloting in the context of a new framework in the national park here called BioTrails, which is in the title of our NSF project. And BioTrails is a series of citizen science events that engage adult volunteers, visitors to the national park, and also locals in collecting specimens and trying to identify them with the help of DNA barcoding. And in a couple more years, hopefully we'll get, fingers crossed, another NSF grant to expand this model and build on what we're learning right now.
That sounds awesome. I always think about this. This would be something good because it starts in Maine, but I've hiked some of the Appalachian Trail, which I love.
Yeah.
But you should set out people who start out at Katahdin. You should give them a kit, a collecting kit. But you could start them out with a kit, and then you could have different universities on the way. And in exchange for dropping it off, you could give them candy bars, ice cream, a shower. You know what I mean?
I hadn't thought of the shower carrot, but I think that's a good idea.
I'm going to hot
little
shower.
hobo bag.
I'm going to make a note of that right now. Shower.
Yeah. Because you know about this, there
are science.
Along the trails there are bear boxes that you can hook your food in, and people leave trail magic or brownies or something like that, and it's amazing when you come across these things.
And there's also this big geocaching thing where people go to certain places and times. Yeah, I mean that's ultimately, and in fact in the proposal I say that we aspire to have this model that we're trying to build here kind of copied and pasted in other national parks and along the long-distance foot trails. So not just Appalachian Trail, but the Continental Divide Trail and the Pacific Crest Trail, because these are outstanding north-south transects, which could be used to monitor climate change and all kinds of stuff. So invasive species and everything else. So that's a great idea, but what I've learned is that projects that have tried this in the past haven't been too successful because they've tried to give people here's a laminated sheet that tells you what to do. Now you go out and do it, and you give us the data. And people just need more help than that. They need to be not completely autonomous, but semi-autonomous. So the idea we're hoping to pilot soon is where you have this kind of slightly more experienced group of volunteers, a smaller number of them, who help guide. They're sort of like docents, and they help guide the majority of your participants, which are complete novices, to do what they're supposed to do. So I would imagine if we use the Appalachian Trail as an example, you might have people who are local to different sections of the trail and who go out once a week or twice a week to find through hikers and make sure that they are doing what they hope that they're doing and understand everything and aren't having any problems and things like that. So that's a layer that we've learned now that needs to be added onto there because completely autonomous work is it's too hard to ask people months in advance to do this, and this, and to have them do it correctly.
Karen, you mentioned earlier that you applied for the Astronaut Program, the U.S. Astronaut Program, and you got pretty close. So how do you go from Darwin and genomics to space?
By getting on a rocket? No. So what happened was, through my involvement in the Beagle Project, I got interested in space again, and here's how it happened. This is the short version, so don't worry, because I know we don't have a whole lot of time left. So in 2007, I got an email out of the blue from an astronaut in Houston who said he read about the Beagle Project in Science magazine, where there was a little news and views blurb thing, and said that he's going to be in space in 2009, and he has an interest in sailing and marine biology, and so he wants to work with me. At which point I screamed and ran around our flat in London and got very excited. And one thing after another, I ended up going to Houston to meet with a team of people there, and we formed a collaboration to combine observations from the space station with, potentially in the future, with ground truthing biological collections from aboard HMS Beagle, or the new Beagle. And during that whole process, we did a number of kind of small demonstration projects together. And when I visited Houston and I saw the rockets and everything, and I met astronauts, and I was just reminded how much I loved space from when I was a kid. And right around the same time, the whole NASA Tweet Up thing got started. And so I signed up to attend a Tweet Up, one of the launches of the last 3 space shuttles. And I got in, and so I attended that, and that was it. Once you see a space shuttle launch in person, you want to be an astronaut. So it's
the giant thing rolling into space on a giant ball of fire is what made you want to ride it, essentially.
Yeah, pretty much. Yeah.
Fair enough.
But also, I think meeting actual astronauts, and he actually phoned me from space in 2009, and my cell phone rang, and it said,
space.
And it says I have an incoming call from 000,000. And I'm
the
like,
country code for space is so weird.
Oh, who is that? Oh, I guess I'll answer. And I answer, and it's like, hey, it's Mike. And I'm thinking, oh yeah, Mike. Oh my God. Because I knew he was in space, and he said, yeah, I'm on the space station. How's it going? And he was asking me all these questions about, he was clearly missing the home comforts of Earth, because he was asking me all these questions like, so, are you going to go down to a pub tonight and have a burger and a beer? It was really good. And I said, this phone call is not going to be about me. We're going to be talking about what you are seeing out the window right now. And that's basically all I'm interested in.
I would have been like, what are the charges for this call?
Well, he called me, so I assumed it was okay. So yeah, so that was very exciting. Anyways, it was Mike Barrett who later suggested to me that I might have a chance if I applied, and also a number of friends on Twitter who I'd met at the NASA tweet-ups said, you should apply. So I did, and I made the first 95% cut, as did a few other people that are out there on Twitter, like Astro Katie and Planet Doctor, Dr. Planet Doctor. And I got the FAA medical exam for spaceflight, and I got sort of along in the process, and then I didn't get invited to an interview. So, but only a couple hundred people did, so I made it pretty far. And maybe next time I will try again, and we'll see how it goes.
That's cool.
They should select based on number of Twitter followers.
I totally agree. But then I think tweets out loud will get it.
Yeah.
If Commander had to,
it's gotta be like, how do you improve your CV to be an astronaut? Like, that's
listen, I thought my CV was pretty good. I was pretty proud of it when I submitted it, and I thought, look, I do all these things, and they're gonna be, and I'm like a mountain hiker and backpacker and stuff, so I can deal with cramped quarters and deprivation and everything. And then when they announced the astronaut class of 2013, and I looked at their actual CVs, I went, oh my God. Because they're amazing people. They really are. They are they're generally in their mid-30s, and yet their CVs are the sort of CVs you would see on a person who's 70. They have 3 or 4 careers in one. Like one woman had done all of this research in marine biology, and now she's an assistant professor in anesthesiology at Harvard Medical School. Well, how did that happen? So she obviously changed careers completely.
She's got a twin. A secret twin. That's how you do that.
Yeah. So that was a little bit of an eye-opener. We'll see. I need to get myself to Antarctica or something. or I need to get
second career starts now.
Or I need to get a pilot's license or something.
All right, space is cool, but before we end up with, before we finish with you here, I got to know more about the Beagle Project because, I mean, I've read The Voyage of the Beagle and I thought it was cool, and I want to know exactly what you guys are planning on doing. Like, are you going to retrace all the steps, all the things he did? You know, I mean, Darwin was a badass in Voyage of the Beagle, right? I mean, he's rolling around in revolutions and. You know.
Yes, he is a badass, and a lot of people don't realize, A, how young he was when he went on the voyage, just 22, and B, that he was miserably seasick the whole time, and he could have gotten on any old packet ship and come back running to England to his rich father, but he didn't. He stuck it out, and he puked his guts out for 5 years, so props. Anyways,
sounds like a PhD, sort of. I don't know.
Yeah.
Yeah.
Yeah, and it was 5 years, so it's about that. Yeah, so what we're planning to do is, so the original Beagle is somewhere under the mud in an estuary in Essex, and I guess theoretically it could be recovered and put in a museum somewhere someday, which I hope actually happens. But our plan is not to dredge it out and rebuild it from that. Our plan is to build a replica, a sailing replica, a modern sailing replica. So not like an exact historical replica, but rather something that looks like the Beagle from the outside, but has a more modern interior with diesel engines, which are required for modern sort of sailing and safety and everything, and enable you to get to ports on time and that kind of thing. And more creature comforts, fewer crew members. There were 75 members of the crew of HMS Beagle on a 90-foot ship. I don't know if any of you have been on a small ship like that, but I don't know how they fit 75 people in there. So it'll be, you know, it'll be a little bit more comfortable than it was for them. And there will be laboratories and more modern facilities on board. But the idea will be that we will provide ship time for scientists, but we will also have on board teachers, students, PhD students, perhaps working with the scientists who are our shipboard scientists. And people will rotate on and off instead of like Darwin being on the ship the whole time. Maybe someone will do a 2-week leg here or a 1-month leg there. And they'll time that to coincide with the areas that they need to collect their field specimens, that kind of thing. So, you know, potentially we could get hundreds, if not thousands of people in the course of a repeat voyage participating in person. Plus, if you add on top of that all of the people who might visit the ship when she's in port in all of these places, and all of the people who could follow online on perhaps a layer on Google Map, Google Earth, that kind of thing, you know, it could potentially reach, you know, hundreds of thousands of people, including students and of all ages, really. So that is our intent.
That's awesome. I would love that. Especially like a classroom could follow one specific project.
Yeah.
That would be amazing.
Yeah, and there would be like live
that's fantastic.
You know, crow's nest webcam and things like that.
Yes, I'm guessing you're not going to do everything they did, though. I mean, like the tortoise cookouts and, you know.
Right. So it would be about using the original voyage as a hook or a guide to repeat. And the idea is that we would visit all of the ports that the Beagle visited and then some. So we might go to more places. Like, as a U.S. citizen, I would really love for the Beagle to visit the United States. So I would love it if we could add the East Coast, at least, of the U.S. To the beginning of the itinerary. But as long as we don't miss the important and even the small ports that were important for the Beagle along the original voyage, I think we can certainly add more.
Cool. Well, I think it's a fantastic project, and I think there'll be a lot of people following along with it. We're pretty much out of time right now, but where can people follow along with everything that you're doing? Because it's clear that there's a hell of a lot of it.
Yeah. Okay, so I tweet at K.E. James about my barcoding and citizen science stuff, plus about a million other topics. And there's also a Twitter account for the Beagle Project, at Beagle Project. And there's a website, www.hmsbeagleproject.org. And let's see. You can find out what's happening at the Mount Desert Island Biological Laboratory at www.mdibl.org. Am I missing anything? Yeah. Mostly on Twitter. That's where I live online. That's kind of my primary spot. And when I do blog, I will tweet about it, so you won't miss anything.
All right, so that's episode 51 of the Breaking Bio podcast. You can find us online at breakingbio.com or on Twitter at breakingbio. I hope you join us next week for another new exciting episode.
Okay. Bye.
Bye.